Under drought stress, we found that the tolerant genotype T400 accumulated much more osmolite, has higher osmolarity than the sensitive one under drought stress. Under well-watered conditions, the leaf osmolarity are similar. So also, besides we compare the shoot, the leaf physiological features, we also compare the root features. We found that in general, the T400 has longer roots than 279. This will contribute to drought tolerance in the field, so they can explore deeper water. So after we did these physiological comparisons, we analyzed the metabolite accumulation and transcript changes under drought stress of these two genotypes. This experiment was done in a greenhouse. We grow these plants in big black plastic cones in soil. And for each cone, we buried a soil moisture meter, so we can monitor the soil moisture level changes real time. And for the stress, we apply 3 stress levels corresponding to soil volumetric water content of 10%, 5%, and 1%, respectively. So at a soil volumetric content of 10%, it's actually the starting of stress. The leaf conductance decreased about half of well-water conditions, and the total experiment takes about 2 weeks for the severe drought stress. So next, I'm going to give you some, show you some results we got with the metabolite analysis. So now I'm only show you the results with the severe stress plants compared to well-watered plants. So this is the GCMS data of the polar metabolites, and it's just showing you the log2 transformed ratios. So if it's red color means upregulated, and if it's green means downregulated. So from this we can see under severe drought stress, in the shoot, the 400 E plus and E minus overall had a majority of metabolites strongly upregulated. However, in the 279 sensitive genotype, most of them downregulated except a few amino acids and sugar. In the root, the situation is overall much similar with the T400E plus. Stress. So the first metabolite I pick is proline, because we found that proline under severe drought stress, it has a specific accumulation only in the tolerant genotype, but not in the sensitive genotype. So sorry, I need to just explain a little bit more because the X panels, there are 4 panels corresponding to well-watered, mild stress, moderate stress, and severe stress. And each panel has 4 columns, 279 plus, minus, 400 plus, and minus. So it's easy to understand. So this is for the metabolite accumulation. And the next, let's look at the key genes involved in proline biosynthesis, is P5CS. And we can see that P5CS also has a higher specific upregulation in the T400, but not in 279. It's higher than well-watered, but it's much lower than the T400, especially in the shoot and also in the root. So this is the first metabolite I picked. And the second metabolite we found is very different accumulation is raffinose. The raffinose, we can see clearly that in the root, there's a specific high accumulation of raffinose only in the T400 and not in 279. And in the shoot, it's more complicated with there are very few accumulation in 270 minus, but some more accumulation in 279 plus. And next, let's look at the genes involved in raffinose biosynthesis. So now I'm going to give you some review for raffinose biosynthetic pathway. And in plant, the 2 major enzymes, one is galactanosensis, and another one is raphinosensis for raffinose biosynthesis. And the raffinose also can be used to further synthesize for stachys, the tetrasaccharides. This is trisaccharide. So this is the whole pathway. And then if we look at the gene expression of raffinose biosynthesis, we can see the raffinose biosynthesis gene, the pattern actually is more or less similar to P5CS for the proline biosynthesis. You see in the shoot it has higher in the T400 and in the root as well. It has a more higher upregulation in T400 overall. Look at it in the T400. And the next, if we look at the galactinosynthesis, it also has a higher expression in the T400 than in the 279. But this expression pattern, if we look at this, it does not match with the metabolite changes because we saw a transient increase of raffinose in the root, if you remember. So wait until we see the expression pattern of stachys synthesis. Because we found that stachys synthesis gene expression were extremely highly regulated and drought stress, especially in T400. You see this, and also in the root. So because stachys is the substrate to synthesize, raffinose is the substrate to synthesize stachys. So from this result, you can see the stachys, the pattern does not match with the genes with those stachys synthesis and galactinosynthesis because stachys was used to synthesize. Raffinose was synthesized to synthesize stachys. So, but the GCMS experiment did not measure stachys. So, but we anticipate stachys metabolite accumulation is much higher in T400, which we will measure in the next step. To summarize what I just talked, so we did physiological comparisons and metabolite as well as transcript comparisons between these two pathophysical genotypes with different drought tolerance. And we found that the tolerant genotype had narrower and thicker leaves, longer roots, and smaller stoma density and transcription rate, and also has higher osmolyte accumulation. And this high osmolyte accumulation can be confirmed with GCMS analysis of metabolites. And also we found the proline and raffinose as top two osmolytes, which had different accumulation patterns between these two genotypes. And the transcript analysis with RNA-seq can highly consistent with the metabolite analysis, but we have to put everything in the network. So you cannot just pick one gene and predict metabolite. You have to put together with with other genes and in the network. So just mentioned just now from the previous talk of Dr. Patel, we also found a lead protein, which are a group of lead proteins are very extremely highly regulated in T400 but not in 279. To finish, I'd like to acknowledge all the people involved in this project. Thanks, Michael, for the consistent support, and also our collaborative lab, Malloy Saha's group, for the field experiment. And also want to mention we have a poster for alfalfa drought stress tolerance from my colleague. Thank you very much for your attention.
Thank you for your good presentation. We move on to the next presentation, which is by Rohit Dhakre, again from Germany. And he will be talking on negative regulation of drought tolerance in rice by jasmonic acid. Evidence is from physiological traits, root proteome, and metabolites. Dr. Rohit, please.